Hello, I'm Margarita Karapetyan, a PhD student from Chromatin Biology Laboratory at the Agricultural University of Georgia. I'm happy to be given an opportunity to present the results of our project, Zebrafish as a model for genotoxic anti-cancer drug research. Topoisomerases are ubiquitous enzymes that govern DNA topology in all living systems. Depending on the peculiar DNA cleavage mechanism, topoisomerases are classified as type 1 and type 2 enzymes, introducing transient single or double-strand DNA breaks respectively. All higher eukaryotes contain a single topoisomerase 1 enzyme and two isoforms of topoisomerase 2, A and B. Topoisomerase is ubiquitously expressed and engaged in DNA replication and transcription by relaxing DNA suprahelical tension raised during these processes. Topoisomerase poisons stabilize covalent enzyme DNA intermediates, covalent cleavage complexes, which are highly cytotoxic, especially for rapidly dividing cells. For the several decades, topoisomerase 1 and topoisomerase 2 poisons are widely used in anti-cancer therapy. To study the effect of topoisomerase poisoning on viability, 1 day post-fertilization embryos were exposed to topoisomerase poisons for 24 hours and mortality was monitored daily for 8 days starting from day 2. For topoisomerase 1 poisoning, embryos were treated with camptothecin derivatives, irinotecan and rubitecan, while for topoisomerase 2 poisoning, both the DNA intercalating agent doxorubicin and the non-intercalating agent etoposide were used. As shown on figure 1, topoisomerase 1 poisons markedly decreased the viability of embryos. However, no significant changes in mortality were observed in embryos treated with topoisomerase 2 treated embryos. Tolerance of 1 day post-fertilization embryos to topoisomerase 2 drugs could be attributed to several reasons. Firstly, it is possible that during incubation of the embryos in medium containing doxorubicin and etoposide, drug uptake is limited due to biological barriers such as chorion. Therefore, to ensure the proper delivery of topoisomerase 2 drugs, we microinjected etoposide and doxorubicin directly into embryo's yolk, and again, no changes in mortality were observed. Secondly, zebrafish embryonic cells might be resistant to topoisomerase 2 poisons, and if so, no significant topoisomerase 2 poison-induced cleavage complexes known to be associated with high cytotoxic effect should be detected in treated animals. To test this hypothesis, we monitored the effects of topoisomerase 1 and topoisomerase 2 poisons on DNA damage using alkaline comet assay, sensitive to both double-strand breaks and single-strand breaks, the hallmarks of topoisomerase 1 and topoisomerase 2 cleavage complexes respectively. DNA damage was measured in isolated cells from 1 day post-fertilization embryos treated with topoisomerase poisons for 1.5 hours. As shown on figure 2, rubitecan and etoposide both induced significant DNA damage in embryos. However, considerable DNA damage was observed also in non-treated animals. In order to determine whether the observed basal DNA damage was due to the existing endogenous topoisomerase 2 cleavage complex, we treated the slides with agarose immobilized embryo cells with EDTA, known to reverse topoisomerase 2 cleavage complexes. As shown in figure 3, the addition of EDTA significantly reduced the amount of damaged DNA, suggesting that the observed high basal level of DNA damage originates from topoisomerase II cleavage complexes. Thus, the tolerance of topoisomerase II poisons could be attributed to the tolerance of topoisomerase II cleavage complexes, or doxorubicin cellular uptake could be different before and after embryo processing for comet-assay. To check this latter hypothesis, we use doxorubicin intrinsic fluorescent properties to monitor its delivery in 2 and 4 hour post-fertilization and 1 day post-fertilization embryos. In 2 and 4 hour post-fertilization whole embryos, doxorubicin fluorescence was detected predominantly in the cytosol, leaving nuclei unstained. It can be seen at figure 4a and 4b, while in 1 day post-fertilization embryos, the presence of doxorubicin was mainly restricted to cell boundaries, figure 4c. On the other hand, in cell suspension prepared from doxorubicin-treated 1 day post-fertilization embryos, doxorubicin fluorescence was detected predominantly in the nucleus, figure 4e. Similar nuclear distribution of doxorubicin was observed in human cell line. Therefore, we can assume that during single cell suspension preparation for comet-assay, doxorubicin translocates from extracellular environment to the cell interior, leading to the formation of cleavage complexes observed in our comet-assay experiments. Previously, it has been shown that poly-ADP ribose polymerase PARP inhibitors enhance topoisomerase I poison-induced cancer cell killing. Poly-ADP ribose polymerase I, PARP1 shortly, is the most abundant and ubiquitous member of PARP superfamily of proteins. Using NAD as a donor of ADP ribose units, PARP1 catalyzes the covalent attachment of poly-ADP ribose polymers on itself and or other target proteins. In mammals, PARP1 controls a wide array of cellular processes such as DNA repair, transcription, cell division, heat shock response, etc. It is well established that PARP1 functions as a DNA damage sensor and is implicated in DNA single-strand breaks and double-strand breaks repair. Bounded to DNA damage sites, PARP1 also helps to recruit and modulate the activity of other nuclear proteins engaged in DNA repair pathways. Also, it has been reported that at the initial steps of apoptosis, temporal PARP activation is important for caspase-dependent apoptosis. It has been shown that in mammalian systems, PARP inhibitors enhance DNA cytotoxicity of topoisomerase poisons. Therefore, we studied the effects of co-administration of PARP inhibitor olaparib and topoisomerase poisons on zebrafish embryo viability. As it can be concluded from figure 5, olaparib alone had no influence on embryo viability. However, olaparib treatment significantly increased the mortality of irinotecan-treated embryos, while etoposide in combination with olaparib did not show any effect. At the next step, we decided to find out if olaparib increases DNA damage triggered by topoisomerase I poisoning. In DNA damage recovery experiments using comet-assay, olaparib increased DNA damage level in irinotecan-treated embryos as seen at figure 5. These results are in keeping with previous data demonstrating that PARP is involved in topoisomerase I-induced DNA damage recovery. To confirm that olaparib was able to inhibit zebrafish PARP activity, we performed immunocytochemical and Western blotting analysis using anti-poly-ADP ribose antibody. As shown in figure 6 and figure 9, PARP inhibitor completely abolished poly-ADP ribose synthesis in zebrafish embryos. It is well established that PARP1 is involved in the repair of DNA damage induced by alkylating agents. To define if the same is true for zebrafish, we incubated 1-day post-fertilization zebrafish embryos with dimethyl sulfate, DMS, in the presence or absence of olaparib and monitored embryo viability and DNA damage recovery. According to the obtained results, PARP inhibition and DMS-treated embryos dramatically increased both the mortality, as can be seen at Figure 6, and the DNA damage level compared to DMS alone treated groups. In order to understand the role of poly-A-D pyribosylation and PARP1 in topoisomerase I poison-mediated apoptosis, we treated zebrafish embryos with different concentrations of rubitecan and tested for the apoptotic cleavage of PARP1 by Western blotting analysis. As you notice in Figure 7, 24 chylidatan PARP1 fragment was detected in embryos treated with 5 and 7 hours, like here, but not in those treated for 3 hours or untreated ones. As far as we know, this is the first time when the full-length PARP1 and its apoptotic fragment have been detected by Western blotting in zebrafish embryos. Thus, PARP1 could be proposed as a specific marker for caspase-dependent apoptosis in zebrafish embryos, as it is established for mammals. For in situ detection of apoptotic cells in rubitecan-treated zebrafish embryos, whole mount immunofluorescence using anti-activated caspase-3 antibodies has been performed, as can be concluded from Figure 8. Caspase-3 positive cells were observed throughout the body of rubitecan-treated embryos, showing predominant localization in the brain, eyes, and notochord area. At the next stage of our research, to evaluate the effect of pyrylation, poly-A-D pyribosylation, on rubitecan-treated caspase-dependent apoptosis, embryos were treated with 1 millimolar and 2 millimolar rubitecan in combination with PARP inhibitor olaparib. Due to the presence of high-density caspase-positive cells in the head area, it can be seen here, we focused on the quantification of individual signals in the embryo's eye. Olaparib significantly reduced the presence of caspase-positive signals in 10 millimolar rubitecan-treated embryos, but not in 1 millimolar rubitecan-treated embryos. However, olaparib failed to reduce the eye's developmental delay in 10 millimolar rubitecan-treated embryos. Our findings that PARP inhibition in rubitecan-treated embryos causes a decrease in caspase-3 positive cells without affecting developmental delay is in agreement with the previously obtained data from different model systems and clearly indicates that PARP1 and poly-A-D pyribosylation regulates the cell death pathways in zebrafish embryos. That is all for today. Thank you for your attention.